Jennifer Doudna. She moved to Hilo, Hawaii when she was 7, and as a non-native family, she felt very alone. She used books to cope with her feelings and to learn about the world around her. She graduated from Hilo High School in 1981. After her freshman year in college, she doubted herself in the scientific field and had thoughts about changing her major to French, but one of her professors convinced her to stay in science. She went on to graduate from Panama College in 1985 with a Bachelor of Arts, and then went to grad school at Harvard University and graduated from there in 1989 with a Master's of Arts and a PhD. Her interest in science started with her reading books around the house that her father left around. When she was in sixth grade, her father gave her a copy of James Watson's 1968 book, The Discovery of the Structure of DNA, the Double Helix, which really interested her in science. Some of her hobbies include running, biking, nature walks, gardening, and reading. I chose her because at this time in class we were talking about DNA and all the nitrogen bases, and I thought it would help me learn more about it and make a personal connection so that I can remember the material better. Her field is chemistry, and she is currently focusing on redefining CRISPR technologies. My primary source that I use is her talk at Berkeley, talking about CRISPR and the future of gene editing. She starts off by saying that Jill Banfield called her wanting to talk about the phenomenon that she discovered in bacteria that is RNA-centric. Through Donda's research, she found out that bacteria had the ability to learn about viruses that infected them and store that information in the genetic vaccination card and use that information to protect itself. They found that the same system can be used in animal and plant cells to trigger DNA repair, and in doing so, they discovered they can induce targeted changes into the genome. The reason they can do this is because there's time to fix DNA breaks in plant and animals because they grow slower, unlike in bacteria. CRISPR in human health was also one of her points. She claimed that Stuart Orkin found a transcription factor, BCL11A, which is a protein to turn off the production of fetal hemoglobin, which is the cause of sickle cell disease. She claims that CRISPR could be the cure to sickle cell disease, but some challenges of CRISPR is that it's expensive because it's about $2 million per patient because it's hard to deliver CRISPR into the blood stem cells. Another point she made was into the future with CRISPR. She started this off with the general delivery of CRISPR, not viral versus non-viral. She then went on to talk about her experiment with humanized mice, where they used EDVs with CRISPR-Cas9 and injected the mice once that targeted human T cells. Jenny found, and I quote, that targeted precision genome editing happened only in the T cells in these animals that had been treated with the Cas9 carry EDVs, end quote. Building teams to focus, they want to build teams to focus on research, which allows targeted editing, which could help with the cost of CRISPR. They got FDA approval for a clinical trial for sickle cell disease with CRISPR-Cas9 therapeutic strategies to also help reduce the cost. She wants to work with the FDA to turn CRISPR into a platform, which means it's therapy based on the same protein, and the only thing that would change would be the RNA molecule that guides it into the genome. Figure one is from her speech, which indicates the starting on the left side of the image is the harvest of stem cells from a patient. The harvested stem cells then isolate CD34 plus and add ex vivo gene editing. After the process, the gene is edited and infused back into the patient. Figure two shows that they have an idea to form these two delivery strategies of CRISPR into one, allowing them to work together and deliver pre-assembled CRISPR-Cas9 guided RNA molecules that looks like a virus but doesn't have the downside of an actual infection. Some things that she's uncovered is RNA folding and function, which happened in her earlier career, CRISPR-Cas9 as a gene editing tool, mechanisms of CRISPR-Cas systems, new CRISPR tools and variants. Her next steps involve the solve the delivery problem for CRISPR therapies, scaling CRISPR therapies to a broader use, expanding into more complex diseases, and gene editing into agriculture and climate resilience. Jennifer Doudna has been making incredible pathways in science and continues to do so. She plans to make CRISPR more available for the people who need it. She is an incredibly selfless woman who is trying to help people understand the world around us, including things we cannot see.